Hi, my name is Ronit Langer, and I'm a junior studying electrical engineering and computer science. And I'm really interested at problems that exist at the boundary of computer science and biology. One of the really pressing problems that I'd like to address in my research is the growing opioid crisis and the effect that it's having on our first responders. When a first responder comes onto a scene, they're not sure what's in the air. And the problem is that more powerful opioids, when they're aerosolized, could be potentially lethal. And so what we want to do is create a biological sensor that's able to tell the first responders whether or not those opioids are present in the air. The problem is that the current biological sensor we have available to us is the natural mu opioid receptor that lives in our brain. But the mu opioid receptor actually binds promiscuously to all sorts of opioids, leading to the opioid crisis in the first place. And so what we want to do is to take this mu opioid receptor and mutate it in such a way that it binds very specifically to powerful opioids. The problem is in protein engineering in general is that we don't have a good understanding of the relationship between sequence, structure, and function of a protein. Also, the possible sequence space that exists when you have 20 amino acids is so much larger than the observed amino acid space that we see in nature. And so what we want to do in this project is to be able to take the mu opioid receptor and mutate it in silico, meaning on the computer, and then build it in vivo and test it in living cells such that we can speed up this process and create more powerful protein sensors. And so the way we do this is we curate a large training set of very similar proteins to the mu opioid receptor, and we test to see how different mutations affect them in living proteins. And then we use that to train a machine learning algorithm, which can then recommend better sequences for us moving forward. And then we go and we test those sequences in cells, and then this process iterates until we get to the desired functionality of our protein. And so once we've done this for the mu opioid receptor, we can take this technology and extend it to many other problems that exist in the protein engineering space.